And the agenda for today is that we have, I'm now giving you a brief background of a few words on the two softwares that we will use for this demonstration, and also a background of the data set that we're going to work with initially. And then we will do the actual use case demonstration, showing you how we can use our two softwares and use them in a common workflow during the data exploration. And we will end with a Q&A session where you have the chance to ask questions. So we will use the chat for the questions. So please, if you have questions, write them in the chat. We are two companies with two different softwares. So Qlucore, which I represent, our software is called Qlucore Omics Explorer, and Nebion, that Anneli represents, have a software called Genvestigator. And the two softwares, the challenge that many of you face today is that the size and complexity of many of the data sets that you work with really distances biologists from the data. And the other challenge is that there are tremendous amounts of published data. So with the combined use of our two tools, we can meet these challenges, and we will show you how that is done in this workshop. So Qlucore Omics Explorer, it's an easy-to-use tool to find and visualize relevant genes within your data set. And Genvestigator will help you to compare expression patterns from your data with thousands of transcriptomic studies. So by using both the softwares, you will be able to explore public transcriptomics data sets with visualizations and enriched annotations. The use case that we will demonstrate is how to do a data set exploration and subsequent study finding. And the data set that we will use for this is a data set on IBD enriched with annotations from Genvestigator. We will import this data set in Qlucore Omics Explorer, where we will explore, analyze, and visualize the data and annotations. And at the end of the session, also create a gene signature. And this gene signature will be exported to Genvestigator. And then Anneli will show you how to use this gene signature to identify similar data sets and also new indications in Genvestigator. And she will then pick one of the new studies that she finds and export this back to Qlucore Omics Explorer for a deeper exploration and analysis. So you can work with these two softwares in a loop. You can go back and forth between the tools. A few words on the data sets. It's an infliximab treatment of IBD. So it's based on a geo data set. You have the number here. And the title of this is Mucosal Expression Profiling in Patients with Inflammatory Bowel Disease Before and After First Treatment. So in this data set, we have samples from patients that suffer from Crohn's disease as well as ulcerative colitis. And these are autoimmune diseases characterized by chronic relapsing inflammation of the gastrointestinal tract. And the infliximab, it's a TNF inhibitor, a monoclonal antibody against TNF-alpha. And this is a common treatment for autoimmune diseases, for instance, rheumatoid arthritis, Crohn's disease, ulcerative colitis, and psoriasis. And the experiment, the samples have been analyzed on an Affymetrix microarray, and in the data set there are 131 samples from 73 subjects, 61 patients suffering from either Crohn's disease or ulcerative colitis, and 12 controls. So we have 37 Crohn's patients and 24 ulcerative colitis patients. From the Crohn's patients, there have been samples from both the ileum and the colon. From the ulcerative colitis patients, there have been samples taken from the colon only. And these patients have been studied before and after treatment. And they have also been divided into responders and non-responders. And then there are 12 control samples from normal tissue from both ileum and colon, and that has not had any treatment. So now I will move over to the demonstration. And so I will leave this and just go over here. And here we have a data set that has been exported from Genvestigator into a Qlucore-specific data format called GE data format. So when I just double-click on this file, it will open up the software and load the data set. So I will just close this little like that. So here we are. So this is one of many data formats that we support in Qlucore. We also support RNA-seq BAM files, cell files, text files, and data set from Gene Expression Omnibus. But now we have loaded the data set, and down here we can see that we have 131 samples and 23,000 genes. And we look at, so each blue dot here is a sample. And we are now looking at the samples in a PCA plot, a principal component analysis, which is an excellent way of looking at your data in 3 dimensions. This data set, of course, has many, many more dimensions, but we look at the 3 dimensions where we have the most information, and this is very useful for them. We can actually visualize our data. And looking at the data here now, we see that it divides up in seems to divide up in 2 groups. And we can even try to remove some of the noise in the data set by just filter a little bit on variance here. So I now removed some of the variables here, some of the genes, and then we can see that the 2 groups, we can see them very, very clearly here. So we would, of course, like to understand what these 2 groups are. So let's have a look at the annotations. So here in the Samples tab, we have a lot of enriched annotations that have been enriched, curated by Nebion. And let's try the anatomical parts annotation here. So here we can see that samples have been taken from the colon as well as the ileum. So let's color on that. And then we immediately see that the 2 groups that we see, they are really the 2 different anatomical parts. So that's a very, very strong signal in this data set. So there is a big difference in expression between, depending on where the sample has been taken. So let's open up a new synchronized plot. So another PCA plot that we just put side by side. And let's color this plot on the pathological condition. So we have the red samples are the samples taken from the Crohn's patients. The green samples are samples from ulcerative colitis patients, and the blue samples are the healthy controls. And here we can see what we already know, that The Crohn's patients, there we have samples both from the ileum as well as from the colon, whereas in the ulcerative colitis we only have samples from the colon. So for my continued analysis now, I would like to focus on only the Crohn's samples, and I would also want to focus only on samples taken from the colon. And looking down here, you can see that I now took away 90 of the samples. I only have 42 samples left. So that can be done on the fly directly. So you can focus on what you're interested in one specific analysis. And now I would like to color the remaining samples on the stimulus annotation. And here we can see that I have untreated responders, I have treated responders, and I have untreated and treated non-responders, and then I have the healthy controls. But the analysis that I want to do now, I would like to focus only on the responders. So let's take away the other groups here for the moment. So now I only have 3 groups left. So the light blue samples here are the untreated samples. The gray samples are the treated samples, and the blue samples are the normal controls. So then I want to do an F-test or Anova test. We call that a multi-group comparison on the groups that I have active now. So I just now, by dragging the statistical slider now, like that, to a Q value, a false discovery rate of 0.01. We can even go down a little bit further. That leaves me with 1,500 genes that are good, very good at separating these groups. And you see that they help us create 3 very clear groups here. And what we can see is that here we have the untreated samples, here we have the treated samples, and here we have the normal controls. And we can see that the treated samples are moving towards being more normal, which is interesting. And let's also have a look in a heat map. Like that. So we open up a heat map and we cluster it on the variable side like that. And we color it according to stimulus like that. So here we have the groups. So here we see the same thing. We see that the normal samples, they have some clear upregulated genes and some clear downregulated genes. The untreated samples have the opposite expressions. So here we have downregulation. Blue means downregulation and red means upregulation. And here, if we look at the treated samples, we see that they are becoming more red. So they are changing from blue to red. And down here we can see that they are changing from red to blue. So that gives us the same picture as what we saw here. We can also see, if we turn this a little, that we have one gray sample here. So we have one treated sample that behaves very different from the other treated samples. So let's use the mark here to see which one that is. So that is this one in the heat map. And that makes sense because this has this more or less the same expression as these untreated samples. So maybe this is a non-responder that we think is a responder, but maybe not. And just a comment, the heat map is very interactive. So if we continue to filter here, you will see that the heat map will just update, and now we see the gene names here. Okay, so let's now save our results. So I have my The genes that I found here, I have them here, so let's just make a copy of this list and call that F-test, like that. And let's also add the Q value, the false discovery rate, and let's sort the list on that, so I now have the best genes on top of the list. And now maybe I would like to have a closer look at some of them, so I will open up a new synchronized plot and make that a box plot. So let's make it a box plot where we have stimulus on the x-axis, and we pick one or a few of the best genes here and show in the plot. So here I now see this gene, and maybe I would like to show the individual samples as well here. Okay, and if I would like to have a look at several genes at the same time, I can do that as well. And if I want to go back and show everything, I can just tile the plots. And all of these plots are very easily exported in a number of different file formats. So it's very easy to get both the list as well as the plot out as files from the software. Okay, so now I would like to find a gene signature. And then I would like to focus on only the treated and untreated groups here. And let's change the heat map here to a scatter, to a volcano plot, like that. And let's color it on fold change, like that. And let's go back a little bit here, like this. So here you see a typical volcano plot. And here we have the upregulated genes, and here we have the downregulated genes. We are now comparing the treated samples with the untreated samples. And again, I would like to do statistical filtration here. So let's go down to a Q value of around 0.01. Here we are. I can even type that in if I want to, like that. And let's also filter on fold change. Let's say that I'm only interested in samples with a fold change of 2 or more. And then we see that I find one upregulated gene. Let's label that to see which one it is. Here we have it. And maybe I also want to label some of these up here, like that. So this is also one way of viewing your genes. And of course, I can also go back and make it a heat map. And then we see the same. I have one upregulated gene and then mostly downregulated genes here. And let's save this as a list. I just make this and call that gene signature, like that. Signature, like that. And let's also add some more things to the list here. The difference between the groups and the fold change, maybe. So I now have that information here as well. And I would like to sort the list on the difference here. And now I can just right-click here and copy the differences. So this is the gene signature that I can then paste into Genvestigator. So then I just go into Genvestigator. I go to the signature part, enter signature, and I just copy paste here. And I have now added the signature into Genvestigator, and now we can continue the analysis in here. So now I hand over to Anneli. I will just make her the speaker. So bear with me just a few seconds here. So, or the presenter. So and for some reason, can you hear me, Anneli?
Yes, I can hear you.
So for some reason now this doesn't work. I'm dragging the little so let's see here. We would like to make you the presenter.
All right. Can you right-click on me and make me presenter, maybe? So far, just a normal participant.
Change role to presenter. Okay, yes. Now.
All right.
Now,
Perfect.
yeah.
There we go.
Yeah.
So I'm going to start to share my screen here. And actually, thank you very much, Anne, for handing over and for making that nice signature in Qlucore Omics Explorer.
You are very welcome.
So before I move on with that signature, I'll give a very brief background to Genvestigator because I think some of you might be new to our platform. And what we do is we make gene expression data sets from public repositories available. Let's see, I'm going to move that one. So we make gene expression studies available for compendium-wide analysis. And Genvestigator can be used to compare your own data sets, set them into context, but also to profile your gene lists and targets across tissues and diseases. You can find co-expressed genes or specifically expressed genes, etc. And what we do is, here you see a little bit of an overview of our pipeline. We download the raw data, we process it, quality control, and normalize, and then we integrate that into Genvestigator, into our database. The annotations of parameters, as well as the descriptions, they're enriched and they're standardized. So all this data can be queried and compared in the Genvestigator tools. And so you have a lot of information about each data set behind all the searches that you do inside of Genvestigator later. So now to our case study. We've explored a data set in Qlucore Omics Explorer, and we've identified a signature of genes that are related or regulated upon infliximab treatment. And this is in responsive patients of Crohn's disease in colon samples specifically. So however, if this is really specific for Crohn's disease, we don't know because we've only looked at this one study. So I'm going to pose two questions that we want to answer next. And it is, using the signature that we created, can we identify more studies with a similar gene regulation? And second, can we find potential novel indications for our drug? So in this case, infliximab. With that, I'll switch over to Genvestigator, the interface. And to start our analysis, we always want to, as a first step, go to the data selection. So out here, we choose what data we work on. I'm choosing the largest Affymetrix platform for human, U133 Plus. And it's loading all the data sets, 60,000 samples on this platform to allow for compendium-wide search across tissues and diseases. Then if you want to look at a particular gene or gene list, you can load that here in the gene selection. What we want to look at is the signature that we received from Qlucore. And for that, we go to similarity search tools down here called signature, just like Anne showed you. And here, I've received the signature, and so I can also just enter that here by pasting it, the list in here, and we'll put the right scaling of the data. So what is in Qlucore called differences is here called a log2 ratio, which is the same measurements actually. And here it's relative value. I'm going to rename this to infliximab treated Crohn's. And then it finds me all the genes, and it loads up my signature here, and you'll see all the values. So in this case, downregulated is green and upregulated is red. So we mainly got downregulated genes from Qlucore. So we've set it to the most similar, and what the signature tool does is searching for as similar profiles as possible. And we're getting the results when we click run here. We get the results completely based on the values of expression, so the expression regulation. It's very transparent. You see exactly how similar these expression profiles are in their up and down regulation. So the results that you see here, each row is a comparison between two groups of samples. So in this case, many of them are treated responsive IBD patients versus untreated. And you can get more information about each row here by placing the tooltip on top of that study identification. And so that we see so many IBD studies here means that we could identify several comparison sets that gives a similar response to the data set that we explored in Qlucore Omics Explorer. That also includes the same drug and the same diseases. Now, if we scroll down a little bit, we'll see that they're not only the same disease, but we also see here a study on rheumatoid arthritis, which has been treated with a monoclonal antibody against IL-6 receptor, which is a similar type of study, but it's actually a different disease than what we were looking at before. Now, the second question was, can we identify novel potential indications for our drug or studies on diseases where this treatment could also be applicable? And a way to approach such a question is to look for the opposite profile to the treatment response signature, since that would give the gene expression profile before treatment or the reversal in disease versus healthy. This is what we also saw when we looked at them in Qlucore. And so this you do simply by changing from most similar to most different. And it brings you the signatures which has the opposite profile. And amongst these results of opposite profiles, we find several data sets on the top, being comparisons of IBD, disease versus healthy. This is what we would expect. What you will also find are studies on skin transplantation, or going down, another one on skin transplantation. We find comparisons on split thickness skin grafting, mucosis fungoides, a thermal injury of skin, etc. We also find further down our rosacea studies on autoimmune disease of skin as well. And so, as you might remember from the introduction, infliximab is also used as a therapy in several dermatological conditions, mainly autoimmune ones, and also against rheumatoid arthritis, which we also find here in the list. An interesting observation that we can also find further on the top is kidney disease. So kidney cancers, actually we find two different subtypes of kidney cancer from the same data set here. So this is actually the value of the compendium-wide searches. And it demonstrates that the importance of sample diversity and integrating samples from various organs, cell types, and diseases can bring you a lot of information just from one single signature that you have. So there are a couple of ways to save these results of the analysis that we just performed. So one is to go, if you want to continue with these same samples, these same experiments and studies that we're looking at in the list, and you want to continue with those inside of Genvestigator in other searches, you can make new samples. And this creates a new data selection here on the side with all the samples, 465, included in all of these perturbations. A second way is if you want to present this to colleagues, you can export the data or the images up here at results and move on with it outside of Genvestigator. Well, another way is that when we identify interesting data sets in Genvestigator, in a data-driven manner like now, we might want to dig down into those specific data sets and find out more because they're new to us. And so in our case, we found that these two kidney cancer subtypes with an IBD-like expression profile, what are these subtypes? Which study do they come from? Are there other kidney subtypes included, etc.? We want to find out more about these particular studies because they're interesting and new to us. So we register here the ID of that particular study, and we can load that study in particular from the data selection. So we go to keywords, we're searching for this study, and we can load that one by itself and study that one further. So it also comes up here in the data selection, and it's only one study included here. So I'll go back to the overview and enter a condition search tool, perturbation, where I can visualize one study or several studies and look at all the different perturbations that you have in here. So we see this is one study with all different types of subtypes of kidney cancer compared to normal kidney tissue. And we'll see all the profiles of the same genes that we were just looking at in all the different comparison groups. Now, exploring data sets in details, that's something which Qlucore Omics Explorer is really good for as well. And so if you want to move on with this list and go and visualize it in a PCA plot, for example, you can actually export that data set from here. So you go to Data Sets, Current Selection, you go to Qlucore, and here you can export your data set as a GE data file. And this is the Qlucore data format, which directly opens Qlucore when you click on it. So that takes a minute, so I'm going to skip that set, and I've actually prepared that exact same data set. And you see it has the Qlucore logo here. And once I open that GE data file, you'll see that here we have, again, a data set. We have all the different annotations about these samples. To the right, we can look at anatomical part. We can look at neoplasms and see here are all these different types of kidney neoplasms. And we can color our data set and keep working with it back as from the beginning of this webinar, we can explore this data set in Qlucore. So this all demonstrates a direct path between a compendium-wide analysis tool with deeply curated database to an interactive statistical tool for visual exploration. So this brings me to the summary of all of what we've showed to you. So we started off with a data set on IBD, which was enriched with annotations from Genvestigator, and then Explore this data set and annotations in Qlucore Omics Explorer. She found some strong differences between tissues. She found that the expression was partially restored to normal upon infliximab treatment of Crohn's disease, and she created a signature of infliximab-treated Crohn's patients versus untreated. This signature was then used in Genvestigator to identify similar data sets and new indications, such as ulcerative colitis and Crohn's disease. We found several studies, as well as studies on skin transplantation, skin injury, and kidney cancer, which we could export as a data set and analyze deeper in Qlucore Omics Explorer. So this has shown you how Qlucore Omics Explorer and Genvestigator is an end-to-end solution for gene expression analysis. It goes from deep statistical analysis of a single experiment to a meta-profiling across hundreds and thousands of samples from the public studies. And this brings a synergy of options to your data analysis. With that, we would like to thank you for your attention of today. We're very happy that you joined us. And if you have any questions, we're happy to take them now, or we can look into what you've already written. So, Anne, do we have any questions? Anne, did we receive any questions? I do not hear you. Not sure if
sorry. Yeah.
Ah, there we go. Perfect.
Yeah. So I'm
Thank you.
encouraging everybody to, if you have more questions, please use the chat to communicate with us. So the first question here is Genvestigator limited to human studies, or can it handle other species?
Right. Yeah, that's a good question. So what we showed you now was human studies, but it also contains data from a lot of different mouse models, for example. It has a lot of mouse studies in there. It also has rat studies, quite some tox studies, for example, and a couple of other organisms as well, model organisms. But mainly, most of the data is human and mouse.
Mm. And then we have another question: how to input or format the clinical phenotypes in Qlucore Omics Explorer? Yeah, so it's quite easy. Either you already have it in your, if you have your data set as in an Excel file, for instance, then you just add it as pure text per sample, the information that you have. You can also add annotations to an already existing data set. So it's all a text-based. We will pull in whatever information you give us. So it's very, very straightforward. I hope that answered the question. And then okay, and then we have another question. How do you import your data to the Genvestigator in the first place?
Mm-hmm. So that's also a good question. So actually in Genvestigator, what you're seeing, all the data sets that you see when I'm showing them here and all the samples that I mentioned, these are from publicly available data sets that we've, the team at Nebion have curated and integrated into the platform. So a way to add your own data is either, as we've showed you with a signature, where you can enter like a differential expression profile or any other type of signature, or you can provide us with the data that you're interested to integrate, and we will process that in the exact same way that we process all the other studies, and we can integrate that for you on a private session on a private server to visualize together with the public data.
Yeah, so I think that also answers the follow-up question that the same person had. How do you combine your experimental data with Genvestigator data? I think you just
I hope that answered both.
Yeah, yeah. Great. So let's see what we more have. With the Qlucore software, is it possible to choose the annotation by yourself, or do we have to use only the annotation already set? No, the answer is that you decide the annotations. We have no already set annotations, so you put in your own annotations to the data set. And then we have one more question regarding trials. Is it possible to try out the softwares? And the answer is yes. For the on the Qlucore side, you just go to the Qlucore homepage, Qlucore.com, and register, and then you can download a 10-day free trial with full support from us. And what about Genvestigator?
Yes, it's very similar actually. So you go to genvestigator.com, and there you can sign up and download the trial version. We only get 7 days, but you get full support. You get access to all the tools. What I should mention is you don't the exporting of data, like we showed now to Qlucore, will not be included in that trial. Because that requires a special license, but everything else and all the tools and all the data will be available.
Great, and I have one more question here. Does the process of our own data by your company, meaning Genvestigator and Nebion, have a cost?
To integrate your private data We do have a cost, yes, because this is something we host on a server, and the work to integrate that does have a cost.
Great. So I think that covered all the questions here. I can't see that there are any more. So yeah, so always very nice to receive a couple of questions after webinar. So thank you very much, and thank you very much for participating. Anneli, I think we will also send out a link to the recording of this webinar, or is that what we normally do?
Definitely. So we'll have this recorded, so we'll send it out. And if you have any further questions about licensing or the tools or specific use cases, then please just shoot us an email. You have both of the addresses here. And yeah, thank you very much for attending, and I wish you a great day.
Yeah, thank you very much. And I wish you the same. Thanks a lot.